Good morning, everyone. So today we have with ourselves Professor Josh V. Paramvel from IIT Patna, and he is our today's expert speaker. So he is assistant professor in the Department of Chemical and Biochemical Engineering at IIT Patna, with a joint PhD from National University of Singapore and Imperial College London, and he has earlier degrees from Imperial College and Kerala University. His research encompasses separation processes, crystallization, and process development, particularly in biotech, pharmaceutical, and sustainable processing domains. He leads the Chemical Process Development Laboratory at IIT Patna, which works on downstream separations, continuous processing, and related industrially relevant challenges. So, welcome, Professor Parambil.
So maybe we'll start. Screen, or maybe I'll just keep it here down.
Yes, both you can keep.
Yeah, that's fine. So you don't want that.
They want me to turn this around.
Oh, don't want to show the Milton. Okay. Okay, so good morning all. So we will, as discussed, we will start with protein crystallization today, and we will talk about, since this course is about alternate proteins, crystallization can come as one of the processes which might be involved in some stages of the identifying or developing alternate proteins. So my focus is on, say, I am from the chemical engineering department, so my focus is on crystallization process development as such, not exactly on the protein structural development, but this serves as a tool in order to identify and characterize proteins as well as separate. So I will take you briefly through the overall the landscape of protein crystallization, why it is important, what are the techniques which are often used for that, and so on. To give a brief background about myself, so I am faculty at the Chemical and Biochemical Engineering Department at IIT Patna, and our focus, especially my lab's focus, is on the separation processes. Primarily two types we are looking at, majorly crystallization as well as extraction. And in crystallization, the major focus is on small organic molecules which are often pharmaceutically relevant. We are also looking at a few inorganic molecules which are kind of energetic molecules, and we have looked into a little bit about macromolecules, including biomacromolecules and proteins. So I started my crystallization experiments in fact during my master's time with protein crystallization. So from there I moved on to smaller molecules because of various reasons, but and because it is much more widely industrially accepted for the small APIs. When you look about the pharmaceutical drugs or the drug industry, almost 70% of the entire drugs which are produced undergo through multiple stages of crystallization. So, that is how relevant or important it is for the pharmaceutical industry. When you look into the agrochemical industry as well, almost all the solid products undergo crystallization at one point of time. And when you look into food industry, there are many cases where crystallization might be important, there are many cases where you do not want the crystallization to take place, or when you have crystallization you want to occur in a specific ways. For example, if you want crystallization in, let us say, ice cream kind of products, you want it to crystallize, but at the same time you want all the crystalline materials to be of very specific size or small size, so that they do not leave that sort of solid feeling in your tongue. And also maybe you want to control how and when they crystallize, as well as when they melt. For example, you might be, you might have tasted different companies ice creams or different companies chocolates, etc., which gives a different mouth feel, which is typically related to their crystal size, crystalline properties, and so on. So, we will talk a little bit about that later on. And in our lab, we also do on phytochemical extraction, looking at macro molecules as well as small molecules, and we have focused on developing solvents as well as sustainable processes for identifying as well as separating out and purifying specific proteins or specific bioactive molecules from phytochemicals as well as from mushroom products and so on. So, that is the overall background about ourselves, and considering our expertise in crystallization, as I told you, since we are focusing on the crystallizer development itself. So, we have, for example, this is one of the continuous crystallizers that one of the PhD scholar has developed. So, it is like overall a 10-meter long crystallizer, which acts continuously. So, you continuously feed in the materials and you continuously take out the materials, moving away from the conventional batch systems where you have, let us say, 100-liter vessel or 1000-liter vessel, which continuously like not continuously, which works on a batch to batch basis. So, this is the size of, for example, it is like a few millimeters thick, this crystallizer. When you think about it, it is just a normal tubing. But how do you control the crystallization? So, this is something which is called as a specifically slug flow crystallizer, where you have got the crystallizing solution in droplets, which is being carried by another medium. So, in this case, this is I believe it is a silicon oil which is carrying the crystallizing solution, and the crystallization occurs inside these slugs, and by changing the flow rate, by changing the temperature, concentration, etc., we can modify the crystallization behavior, we can produce specific size of crystals, we can get a specific purity, and several parameters we can change by controlling various parameters, operating parameters on the crystallizer. So, that is one of the studies, and this is a continuous crystallizer. We also work with conventional batch crystallizers ranging from a few, let us say 2-3 ml scale to this is a 2-liter scale vessel. So, trying to work with larger scale, and how does the crystallization change when you are moving from a very small scale to larger scale. So, which is a conventional scale up problem which is faced in the chemical engineering or the chemical industry. We also work on something which is called as co-crystals in pharmaceutical sector, where when you think about crystals, they are generally pure single component systems, but nowadays we are coming up with crystals which contain two molecules embedded within the crystal lattice or the crystal spaces in specific arrangements and orders. So, these are not single component systems, these are multi component systems. So, it is still pure in terms of, let us say, crystalline lattice and arrangements are concerned, but rather than containing one specific material molecule, it contains two or more molecules, typically two molecules. So, this is something like a binary system, and in order to figure out how it crystallizes, under what conditions you would get the stable co-crystal, etc., we produce this sort of or we study this sort of ternary phase diagrams. So, in one axis you have got the one of the drug, say in this case it is sulfamethoxine, in another axis we have got second drug, and this is the solvent system. So, all three together produces single component multi component crystals which are called as the co-crystals. So, it is also an emerging field in pharmaceutical crystallization, which we are working on, trying to develop how do you control those production of those crystals, and how you can optimize its performances and so on. So, that is our expertise on crystallization, and overall I would skip this outline slide because I was expecting to write on here, but overall to discuss about the outline of today's lecture, I will give a brief glimpse on what protein crystals are, where they are useful and how do you produce them, and maybe at the end we can come up with some ideas on how useful they can be in developing alternative proteins. So, to start with protein crystallization, I mean, has anyone seen any proteins crystallize? No. Okay. So, just to give a these are various different types of proteins which have crystallized, and these are some of them are lysozyme, insulin, thaumatin, and so on. And what brings to your mind when you say crystals? What do you feel crystals are? Anything? Sorry? It has got a very rigid or specific structures, yeah, molecular wise or like atomic level when you go about it. They have very specific structures where very specific arrangements and so on. So, that is the first and foremost thing which you notice about any crystalline material. And this molecular level or atomic level structures also gives rise to macroscopic specific morphology. So, these morphologies that you are seeing, some are cubic, some are rhombohedral, some are long needle like, all of these come or arises from the atomic level arrangements that these molecules take or atoms take. So, typically when you look at most of the powder materials that you consider, for example, you take sugar, salt, or even the talcum powder that you apply, most of the time they are small crystalline material. So, it might be surprising when I say that the talcum powder is crystalline, because it feels like very powder, it is not very, it's not rigid or solid like, but when you zoom into it, they are basically very tiny crystals, crystalline particles which are arranged at specific orders and shapes, and that is what it gives the like smoothness. Sometimes it is what gives the consistency between the batches and things like that. Amorphous material, there are plenty of amorphous solid materials as well, but as you rightly said, the crystalline materials major difference is that they have got very specific arrangements. In amorphous materials, they lack that sort of specific arrangements, which might lead to several issues in many materials. For example, when you think about the stability of the materials, crystalline materials, since they have got very structured, ordered arrangements in the molecular level, they are typically bonded much more strongly, they have got better stability in that sense. So, if you want to store something for very long time, especially in the industrial or production processes are concerned, you prefer the crystalline material rather than amorphous material, because the crystalline materials are more stable, they can be stored for a long time without any conversions or physical changes, etc. So, that is one of the major applications or major uses of crystalline materials, whereas in amorphous materials they might sometimes for example, if there is a little bit of hygroscopic or reactive molecules, etc., they have a little bit more of energy, which makes them a little bit more unstable. So, that is one of the major differences between crystalline and amorphous material. And when you look at conventional crystals, see, for example, how do you produce salt crystals, how do you produce? Sorry, you just take the sea water, evaporate it, whatever is left behind is majorly your salt, that is a solid material. So, in a way what you are doing is you are taking a solution, you are removing the solvent out from it, and the material basically crystallizes it. So, that is one conventional method of crystallization, which is called as evaporative crystallization. So, now coming to macromolecules or especially to the proteins, when you look at proteins, the initial examples that I said, especially sugar, salt and so on, they are very small molecules to deal with, and they have got very little flexibility as far as the molecules are concerned and so on. But when it comes to proteins or macromolecules, what happens is they have got quite a lot of flexibility. They have got when you want to try to arrange them or put them into like lattices, or maybe because in crystalline material they need to occupy specific lattices, or they have to have a particular configuration and so on. So, you are going to constrain these large molecules and put them into small boxes and packs, and try to arrange them in a very structured and ordered way, which by means even when you think about it, it seems difficult. And that is one of the reasons why proteins are a little bit difficult to crystallize, but still sometimes that becomes a necessary in order to crystallize proteins. Can you think any application of why proteins are crystallized often?
Mine it's 3D structure.
Yes. So, the most robust way in order to determine the 3-dimensional structure of proteins or the golden standard is basically using crystallography. And in order to do or analyze it using X-ray crystallography, you need to first and foremost be able to produce crystals, good quality crystals in that sense. So, that is one of the major uses or the use case of protein crystallization. At the same time, it was one of the major roadblocks as well, because many a time it took several decades in order to figure out the protein structure of specific antibodies or immunoglobulins and etc., primarily because we were not able to produce good quality crystals of them. So, this became a bottleneck in many cases, especially in the protein development, and because once you understand the protein structure better, then you can engineer it further. Then you understand, okay, how a particular amino acid is interacting with others. So, maybe you want to change the site in which this interaction is taking place. So, you want to make maybe the molecule or the protein a little bit more rigid or less rigid. You want to make it more soluble. So, in order to do all those sort of things, you need to understand how the protein is folding, how the structure looks like, and how how the overall structure stabilizes itself, so that then you can go back into its amino acid chain primary structure and figure out, okay, where should I insert or delete some of these molecules or replace some of the amino acids and so on. So, this becomes primarily important in order for drug development, for protein engineering, for creating new versions of functionalized proteins and so on. For figuring out enzymes, how do you now we are developing enzymes that can break plastics. Now, how do you come about how do you design a particular protein molecule where an active site would be able to bind to a particular polymer or a polymer chain and make sure that it cleaves at that particular point, right. So, this all requires information at the molecular level, which is often produced or developed by proteins. So, there is a major application why protein crystallization, that area of protein crystallization itself developed. But beyond that we have also understood that once we can produce protein crystals, one thing which we are now able to do is that if you can produce protein crystals in a reliable way with a cost effective system, this can also be used as a purification system. Because as I was mentioning earlier, one of the reasons why pharmaceutical companies or pharmaceutical processes often rely on crystallization is it is a one single way in which you can, like in a one single step method in which you can produce very high quality or high purity material. And in drugs, you typically require very high purity, 99% or 99.9% purity. You want as little impurities as there in order to meet the regulatory standards. And how do you do that? Crystallization gives way to a single step process in which you can do that. So, that becomes a major aspect in the sense cost effective purification. And as I told you earlier itself, once you produce the crystals, one thing that you are assured is that it is pure and it is going to be a bit more stable than your amorphous materials, or maybe even more than your liquid solubilized or suspension systems and so on. So, the stability and the purity of the material is assured once you are able to crystallize it, which can have long term impacts in processing, which can have long term impacts in how you formulate particular products and so on. So, I would say that we are yet to achieve into this sort of sustainable crystallization process for many proteins. Some of the industrial ones are being crystallized now, we will see a few examples, but this is a kind of an ultimate goal that we are having. If we can in fact go ahead and start crystallizing many different proteins, that can make them much more cheaper, easily available, and we can even think about taking specific proteins and using it for much more newer and novel applications as well. Now, why this is, as I told you, this has been in the development for more than a century now. but why we are yet to achieve that, there are many technological challenges, and one of the major challenge comes from the nature of protein molecules itself, that as I told you earlier, these are very large flexible macromolecules, and you are trying to put them into a very structured ordered arrangement, putting them like chaining them down into a particular arrangement, which from the molecular perspective it is going to be difficult. And that is where means we or engineering developments often look back into how the cells biology work, because when you look at the any biological organism or even a single cell, they work at the molecular level day in day out. That is how everything is done. They pick individual atoms, they get sodium atoms to move from one part of the membrane to the other part. They are working at a microscopic or atomistic or molecular level scales, which we are yet to master in that sense, in many senses. So, that is why we have nowadays developing systems which can go into micro level, nano level and so on, in order to try to manipulate, and which might be more efficient in many ways, but we are yet to master that techniques, and I believe like maybe with further developments we might reach the 10, 20, 30 years, I do not know, but sometime. So, the complexity of factors will soon see that for protein crystallization there are number of factors which affect, unlike a small organic molecule crystallization that we have, where only maybe 3 or 4 parameters, as long as you are able to control that, you can get a really reliable crystallization. But in terms of protein crystallization, the number of factors increases significantly, which increases its complexity as well. And another major challenge that we have is low feed concentrations. When you are producing small organic molecules, typically through reactions, you can manipulate the reactions, or you can control the reactions in order to produce large tighter values or volumes and as well as concentrations. So, you can get maybe 10, 20 grams per liter without without any major challenges. But when you are producing it through cell lines, when you are producing it through fermentation process, or maybe extracting from plant or animal sources, etc., one of the major challenges is that the quantity of protein or the specific molecules that you are looking for are typically very less. The concentration is means if you get 10 mg per liter, or so something of that sort of range, you might say that okay, it is a very successful one, you are producing enough. But from the crystallization perspective, it becomes a very big challenge in order to take such a low concentration and then to produce a really pure material out from it. So, that becomes a big challenge, that the low feed concentrations which is often produced from fermentation or the other biological systems becomes a big challenge. But nowadays we are able to see, for example, there are many materials or many molecules which are produced through fermentation using engineered yeast or other bacteria and so on, which can give you much more higher concentrations, which the cells are specifically tuned in order to produce much higher tighter concentrations. That is good for as far as the crystallization is concerned, and we will see a few examples later on where those sort of materials or say proteins are being crystallized directly from the fermentation, thanks to genetic engineering and other developments which has occurred. Yeah, please. Sure. Okay, maybe we will come back once. So, if you have any questions, like just once the mic is ready, then we will come back to it. So, a few model proteins that we want to talk about, or when you look into protein crystallization, there are a few model proteins that you often see. And the most common ones means maybe you already mentioned that you do not know any particular model proteins, but that crystallizes, but how about proteins which are used for drugs? Any major ones that comes to your mind? Yeah, maybe we will take your question and then. Yeah.
Sir, as our theme is plant proteins, so crystallization is mostly easily available if we can work on animal proteins. We can easily do because animal proteins are specific and their composition is uniform. But in case of plant proteins, the composition is always a mix of different secondary metabolites, polyphenols are attached, and moreover when we extract the plant proteins, they are often denatured using extracting materials or due to the processing condition. Is crystallization possible in plant proteins?
There are a few plant proteins which have been crystallized, but and see, there are many which has been crystallized in a small scale for structure determination process. But in the larger scale, there has been a few. Means I will give you maybe one example in the next slide as well. But as you said, variations in the structure which occur between, let's say, between plant to plant variations or these sort of things do affect crystallization quite a lot. So there is no universal method to crystallize all the proteins, and it is highly specific on the three-dimensional structure of the protein as well, which is a major challenge in protein crystallization. So I would say that there is no absolutely yes or no answer to that. Some of them we are able to do. It often depends on how much effort has been put into development of this. So technically looking at it, most of the proteins can be crystallized one way or the other, but how far, how cost effective it would be and so on becomes a challenge. And most of the proteins, as you have rightly pointed out, that has been crystallized or even industrially crystallized, they are animal proteins. So with with smart proteins or with protein engineering, when you are trying to develop new proteins, I think this is also something which has to be, which if it can be taken care, where the crystallizability of the proteins can be predicted or maybe controlled in some way, in the long run that would be beneficial. But currently, as of now, state of the art technologies, I would say it is not yet able to predict the crystallizability or maybe make sure that the plant protein that you get would be easily crystallizable at any point of time. It's all experimental screening techniques that has to be done in order to figure that out. But once it is done, then the idea is, the overall end theme is, if you can make something crystallizable, that would be much more cheaper and effective and sustainable in the long run. Yes. But any small changes, yeah, again you might have to go back to the drawing board and start over.
Yes, sure, sir. Sir, in plant proteins, extraction and purity is a big challenge. We are not able to extract proteins up to 99%. Mostly they concentrate and isolate. They are having 90% protein, 70 to 90, and rest 10% comprises of many other components that becomes the major hinder for the crystallization. So practically, as I have worked on it, it is quite difficult to increase the crystallization of the proteins.
So when as you said, if you have 70 to 80% protein content of a specific protein, I am talking about, not a mixture, then yeah, theoretically speaking you can crystallize it, but practically speaking, as you said, whenever there are molecules which are very tightly interacting with those particular proteins, because the molecular interactions matter quite a lot in crystallization. The molecules are arranging one after the other into the structure. So if any other molecules are already closely interacting with the protein, they might hinder this protein moving along with the crystal structure. So that is a big challenge, especially in large macromolecular crystallization, and not only for plant protein crystallization, but even for other ones. So at the same time, there has been demonstrated that you will be able to produce pure crystals from a mixture of proteins under specific conditions. But again, it's all case specific. It's all specific to what is the starting material that you are having. Maybe you produce from one batch of plant, and the next season, due to seasonal variations, the mixture changes, composition changes. I cannot right now assure you that it would crystallize again, but there are developments in that sort of, keeping that in mind. Okay, how can you overcome these variations and so on? And as you said, you cannot directly go into a crystallization process. You have to first do the extraction. You have to first do a preliminary separations and purification, and then go into this. That is possible. On the other hand, if you are engineering the proteins, or if you are engineering microorganisms in order to produce the proteins, or CHO cell lines and so on, from that it might be a bit more easier, because you can little bit tune these sort of systems and then better get out the primary protein that you want, and that would be a bit more pure in that sense to work with. Plants, as you, I agree, completely agree with you. It's a mixture of phytochemicals. Even for our simple phytochemical extraction, we sometimes struggle to get the pure components. So that becomes a challenge. Nonetheless, there are a few examples which has been demonstrated. So yes.
Okay, thank you, sir.
Sir, if we are working on plant proteins, when we extract, we get a crude mixture. So from that crude mixture, if we want to determine the structure, say we are going for some purification technique, and we are separating different fractions of protein, and from that we are able to do this 3D structure or something. But how do we determine that the overall structure of the protein is this? We cannot determine it for the crude protein.
From the crude, not directly, but say if your focus is on the protein structure determination, what is often done is that, especially for the plant proteins, you do multiple purifications including chromatographic techniques in order to ensure that there is only one pure protein that you are getting into the final mixture, and then you go for a crystallization, because you might have already come across the other methods like the LCMS and so on, or even NMR spectroscopy. You can get some or very good idea about the protein structure and so on, but or each of them have got their own limitations and like shortcomings. So once you have this sort of let's say purified protein which in a solution, you can crystallize that and get that crystals which are much more stable and take it for a XRD and get the final words or what would be the definitive 3D structure. Of course, again another challenge is this structure that we are going to get is in a solid crystalline form. When you put it into the solution, in the solution it might be having a slightly different conformation as well. So the structure that you are going to get from the crystallization perspective cannot assure you that okay, this is exactly how the molecule would be arranging or behaving in a solution as well. So that is two different things. So that is where the NMR gets into picture, in the sense that you can get a little bit more information on the flexibility or the position of different molecules related to each other. But NMR currently, or most of the common techniques are limited to small proteins, not with larger proteins. But on the other hand, XRD can handle even larger proteins as well. So there is a different techniques give you some slightly different information, and if you want to really understand okay how the alpha helix is oriented or how the particular structure docking location is arranged and so on, XRD would give you very good information or very good assurance for you within that. So from the plant protein, as long as structure is your main concern, you can do the purifications through chromatography and other techniques. Make sure that you have got a very good pure system to work with, excluding the other phytochemicals that you said, and then you go with a particular screening method which we will shortly discuss, and it is highly likely that you would get a few of them in order to crystallize, and once you have crystals of good quality, then you can take it for XRD and do the structural refinement. But as far as the production is concerned, or that becomes a challenge, because this can be done for a structural determination, but maybe not exactly for a product formulation perspective. I hope we have answered.
Sir, are there any additives that help to stabilize the protein?
Yes, I mean for the crystallization perspective, there are we will come across a few materials, but typically speaking, let's say if you want the protein structure to be determined, you want the proteins to be as pure as possible without many other additives, or if you have additives, you need to exactly know what the structure is of those additives, so that then you can predict that within the structure and then figure out what would be the rest of the protein like. So when you have strong molecules which interact or which closely associate with protein, that is sometimes often used as a tool for protein crystallization as well. So maybe there are many proteins which doesn't crystallize easily, no matter what you do. So in such cases, what you do is you put small peptides or small chains of known structures into the solution, which interacts with the proteins and make it a little bit less flexible or more rigid and so on. These sort of variations can make it more crystallizable, and it can stabilize the protein in its orientations, and that can be crystallized and understood. And of course, the thing is when you get the final solid form, it would also have that stabilizer in it. So, you need to figure out where the stabilizer is and then means remove that in order to figure out what is the actual structure of the protein. So, stabilizers are the typically speaking, small amino acids are often used as or amino acid chains, small peptide chains etc. are used as templates, soft templates for protein crystallization, and they help you in order to stabilize in a number of ways. So, you can do that, and sometimes even polymers, for example, let us say PEG and so on, and its derivatives are often used as polymers in order to drive crystallization. There the idea is not exactly to stabilize the protein, but it is basically means when in solution proteins would be always interacting with other ions, with water molecules and so on. So, these polymers are often added in order to interact more with the solution components and reduce its activity, the solution solvent activity, so that the proteins become a bit more of aggregated and stabilizes themselves. So, they find their right configurations. So, there are these sort of additives which can be used or which is often used in almost all cases of protein crystallization. Any other questions? So, talking about the model protein crystals, see when you look into the literature, most often see as I told you, I am working on a crystallizer development exactly, not exactly on the trying to identify protein structure or crystallize newer proteins in that sense. So, when we look into the crystallization perspective from the crystallizer design, there are a few model proteins which are often studied, and the very common ones are lysozyme, insulin, thaumatin, catalase, then you also have the human serum albumin, HSA molecules, and a few more as well. So, I am not putting in the exhaustive list. I just wanted to highlight out of this, for example, thaumatin is the only protein plant protein which is very easily or often crystallized. The others lysozyme, insulin, catalase, everything are primarily animal based proteins. And when you look at the sizes as well, you can see that it might range or it might change quite a bit. So, you can have small proteins like insulin, or you can have this catalase which is quite large molecule as well. And one common thing that you generally find among these proteins which are very easily crystallizable or the model compounds is that they have a few major domains and a number of disulfide bonds which make the molecules a bit more rigid, because as I told you, entropy of all these molecules which are quite flexible makes the crystallization difficult. So, on the other hand, if you are able to limit the entropy or limit the flexibility by strong bonds like the disulfide bonds, which makes rigid structures, it becomes a little bit more easier for the crystallization to take place. Same thing goes with many antibodies, immunoglobulins, etc., which have got much more stable structures. They are a bit more easier to crystallize. On the other hand, if you are working with protein molecules or molecules which doesn't plant molecule proteins or molecules proteins which doesn't have a very rigid structure, it becomes difficult. Then you have to use these sort of stabilizers as you asked in order to make sure that you constrain their flexibility and entropy to some level so that it crystallizes easily. And means insulin would be very familiar to you all. It is often used as the diabetes drug. Now, one of the reason why insulin is vitally prepared and quite cheap in that sense when you compare with other proteins, crystallization has got some part to play with it. Of course, we have developed newer varieties of how much more efficient cell lines and so on in order to produce insulin with higher tighter volumes and so on. But from the process aspect, means if you are using chromatography separations for purification, that is typically expensive because you have to use the protein chromatography A in order to first maybe get the as pure as possible, then you have to use the cation exchange and the anion exchange columns in order to further purify it or remove the other salts and so on. So, there are multiple stages of chromatography which is often quite expensive for many, especially newer molecules. But if you can go ahead with crystallization, you can maybe bypass some of those expensive methods, chromatographic methods, and then directly go into a much more pure solid material directly from there. And after that, you can take the solid material and redissolve. If your final product has to be liquid form, you can redissolve it. But in one single step, you are able to produce maybe 99.9% pure material, which can be directly taken up for formulation. And there are other developments in insulin. For example, you have inhalable insulin nowadays, or patches which you can, rather than giving as injections, you can have patches which you can put onto the skin, which would give you insulin over like several hours rather than as a single shot. And these can help you in order to stabilize. And most of these formulations, the inhalable ones, you need particles or solid insulin of specific size, so that it basically goes into your lungs, it goes into your alveoli, and then stays there and dissolves into your bloodstream over time. So that sort of control requires control on preparation of that solids, means how big or small that crystals has to be. All of those can be controlled through the crystallization, and that is one of the major reasons why, as I told you, insulin, there are different types of formulations that has been developed, as well as why it is much cheaper as well. But at the same time, acknowledging your questions, developing smart proteins or plant proteins, would they be directly crystallizable? Maybe the answer currently is no. Over the time we might be able to develop some more techniques for that, but currently it is not that easy if you are starting with a molecule which is very large as well as very highly flexible, and where the changes are very often, means that might be having changes from the production perspective. So we have already touched this up in the sense the factors which affect crystallization. Tertiary structure of the proteins do have a significant effect on how they crystallize, or will they crystallize at all. See, for example, this is one protein which has got a quaternary system, or these molecules which are being held together, tetramers are being held together by specific amino acid interactions. If you have a system which is symmetric in nature, if you have a system which is very closely bonded with each other, then it would be a bit more easier to crystallize. When you have systems which are kind of much more flexible, it would be definitely difficult to crystallize. So there are surface amino acids. When you have the three-dimensional structure, what are the surface amino acids which are expressed and which are interacting? If you have, for example, glutamate, it is considered that it is one of the amino acids which can reduce the crystallizability of the proteins. So maybe you can remove and make that into some other amino acids, as long as it does not affect much with its other properties and so on. But it is always a trade off. Let us say, means people do not design proteins for making sure that they crystallize. They design proteins for meeting other applications, and whether they become crystallizable or not during that period of time, that is something which has to be thought about, and if it can be incorporated into the design aspect, then that would be beneficial for the overall market later on. Secondly, the solvents that you take, what is the type of solvent? Are you going to use something which has got very highly hydrogen bonding propensities, or maybe you are using a solvent mixture where you can tune the hydrogen bonding propensity, van der Waals interactions, the salt concentration in that, means what is the electrostatic interactions, all of those things, and sometimes including the viscosity of the solvent, especially when you go into the production level, not in the microscopic product structure determination level. So in those levels, that might also, the viscosity of the solvent might also affect how far your proteins can be crystallized. Temperature, we will see one simple explanation regarding temperature and protein solubility. Environment, in terms of, let us say, when you are having the protein crystallizing from a solution, what are the other chemicals which are present in there, and what are their molecular interactions. Typically speaking, we add certain salts, buffer, etc. into the system in order to modify these sort of interactions and drive the crystallization process, in order to maybe change the solubility of the protein and so on. So there are many additives which are often added. Some are for stabilizing, some are for changing the solubility, and so on. And of course, the pH of the systems also do matter a lot. So the pH of the solution in which it is crystallized, that would also affect how much it can be crystallized. I will just finish this one point. And while crystallizing, especially in the larger scale, when you want to think about into the production scale, the environment in terms of the fluid environment, how much shear is being acted, means it is being mixed vigorously or gently. Those sort of things also do affect, because the proteins are very much shear shear sensitive. Sometimes they do help crystallization, sometimes they prevent crystallization. And having localized, because when you are having a large vessel of, let us say, even 1 liter is quite large when you consider protein crystallization, or if you go into 10 liter or like 50 liter vessels, the localized concentration as well as temperature gradients which might exist in chemical engineering or in the small molecule crystallization, we can generally overcome that by providing better mixing or higher shear and so on. But when it comes to proteins and these macromolecules, the trouble is you do not want too much of shear because that might denature the proteins as well. So, then how do you optimize between these? Like you do not want the concentration or temperature gradients, at the same time you do not want any vigorous mixing as well. So, how do you optimize this? This becomes another set of engineering challenge as far as the production is concerned. Yes.
Thank you, sir. Sir, as in terms of plant proteins or alternate proteins, solubility is important parameter or an important techno-functionality which is required for other. But as we increase the crystallization of any substance or any proteins, solubility is compromised. So, how crystallization is important for proteins in this case?
These are two aspects which might look opposing in terms, because you want proteins to be more soluble. For crystallization you want to reduce the solubility so that it would produce the solid form, but at the same time that is where the role of these other salts, additives, pH, etc. comes into picture. Because maybe let us say your application wise you are going to put the protein into a solution or a formulation where let us say the pH is 10. So, there you wanted high solubility at pH 10, but maybe you can develop your protein in order to have a higher solubility at pH 10, but for the crystallization maybe you can go for a slightly acidic pH or maybe a different salt concentration so that it crystallizes first, take it out, purify it and then use it into your formulation which has got a higher, which will give you a better solubility. So, there are ways in which you can overcome this sort of challenge, especially when you have the capability to design proteins, but if you are looking for conventional proteins or which are already existing, then of course you are not altering their properties directly, but you have to think about altering the properties of the environment. So, can you have additives, can you have some other stabilizing agents or pH control or buffers. So, all of these things can be used in order to tune, or even temperature, as we will come in the next slide. These are the other parameters which you can tune in order to have that sort of solubility difference or decrease where it crystallizes. Then you take it back into your application where the conditions are different, where you might be having a higher solubility. So, this can be worked in favor as well, because another challenge, as I told you earlier, is for a good crystallization to take place or a good yield to have, you need higher solubility to start with, because if your protein is only at a very low concentration to start with, it is generally typically very difficult to crystallize as well. But on the other hand, if you can start with a solution which has got higher solubility, typically speaking your yield would be higher. But during the process you manipulate the process parameters or conditions in such a way that you drive from a system which is having a higher solubility to a system which is having low solubility. During this process we can get the crystals. After that you separate out the proteins or crystals and put it back into the solution or the condition where you want higher solubility and redissolve it. So, then that can be a good thing in one way as well.
But the cost of processing will be too much.
It is always comparative in that sense. Say for example, when it is often said that for a new proteins development of say protein A chromatography and other techniques and the resins which are used for those, the recycling and so on, typically speaking the crystallization operating cost is much lower. Development cost might be higher, but so if you are looking for a protein which you are going for a bulk production, mass market, crystallization plays a long term strategy that you can make it much more cheaper in the longer term, and the product would be much more stable and reliable in that sense. But if you are looking for a protein or a system where you are looking for a small scale production, not too large scale or mass scale, then yeah, I agree with you, the effort that you put into crystallization might not be that effective in the long run. So, that is why going back to the example of insulin, insulin is there in the mass market and you need that at the cheapest cost. Crystallization helps you for that, but going for other monoclonal antibodies and so on, which might be very specific, very low quantity production, maybe protein A chromatography and other techniques might be having economic advantage over. So, it is always a trade off between the volume, market economics and other things.
Thank you, sir.
So, as I told you, one method for crystallization involves just cooling crystallization, where you can manipulate or play with the temperature. For example, this is the solubility line in terms, if you consider a symbol, say for example sugar, the more you heat, you can add more sugar into it, right, because it would be more and more soluble at higher temperature, which is often the case for many biological systems or macromolecules as well. So, typically speaking, the solubility, so this blue line represents the solubility. So, that solubility in fact increases over temperature. Now, if you can, what you have to do is, so under this, below this line, it is, this is the undersaturated region, and this would be the supersaturated region. In the supersaturated region, you can have an equilibrium between the solid crystalline protein and the solution, which is in equilibrium with that. Whereas, if you are under this condition at the concentration and temperature corresponding to this point, any region here, you would always have a clear solution. There is no solids which are involved. So, what you can do is, let us say you start with a system which is at maybe 20 or 35 degrees Celsius, and you cool it down from this 25 degrees Celsius or 30 degrees Celsius into, let us say 5 degrees Celsius. As you move, what is happening is, you will start, let us say this is your 35 degrees Celsius here, you start moving along this line, and it would move up to a particular point where it starts crystallizing. So, it would move into the supersaturated region. At some point it would start nucleating a new crystal, and once the nucleation starts, then the concentration of the solution starts to come down, because the proteins are now moving into the solid phase. So, the liquid phase concentration comes down. So, the concentration would come down and meet with the solubility line, and as you keep on decreasing the temperature, it would follow the solubility line and stop when you stop the temperature cooling process. So, whatever is this concentration difference, you started from let us say this level over here, and you are coming down and cooling it up to this particular level. So, this concentration difference, this vertical difference, that is the concentration of the protein or the material which is now crystallized, and something that you can get within the as solids. Now, going back to your questions in the sense of how the solubility. So, if you have a material which has got a very highly sensitive curve, so a steep curve over here where the solubility is highly dependent on the temperature, it would be very good for you as far as the cooling crystallization is concerned, because you will get a much higher yield out from it by decreasing from a, let us say T1 to T2, and that for a given delta T, your slope of the curve would be much higher, which means that you would get a much higher yield of your material. So, that is one way of producing crystals. So, just to give you an example, so we have we did this sort of milliscale crystallization for a few different molecules. As I told you, the model compounds are insulin, lysozyme, HSA, and so on. So, the idea is this is the chamber in which the cooling takes place, and there is a continuous flow. This is the initial temperature change which takes place, and there is a continuous flow that you can provide, and this is in a milliscale. So, we are using, for example, syringes of 10 ml, 5 ml, 2 ml, etc., where you feed the protein solution to start with. So, this is generally at the room temperature to start with, and then at a given fixed rate you pass them through the tubings. These are few millimeter diameter tubings, temperature controlled with an external recirculator and so on. So, as it moves through the system, moves through the capillary tubes that you are providing, it cools down and it starts to crystallize. So, now the thing is, you can have systems, you can control the length of this system over here. You can make it longer or shorter, or you can take it out from here and then again cool it in a secondary chamber as well. So, by doing that, what happens is it starts to crystallize inside this capillary tubes, and you can get the crystals which starts to form inside the tubes. And this study was conducted in order to figure out what is the impact of shear in crystallization process, in protein crystallization. And so, these are different, this is for 24 hours, this is for 48 hours. So, you can see that for 48 hours we can continuously do the crystallization process as well. So, it can be a continuous or a semi batch sort of processes that we can do. And with shear, with varying shear. So, if here you are increasing the flow rate, you can see like from top to bottom, what you are doing is you are increasing the flow rate, which changes the residence time as well as the shear conditions or the velocity gradients inside the tubing. And in this particular case, for example, as you increase the shear, you are getting more and more crystals, which means that the nucleation is being affected, or the development of new crystals, birth of new crystals are being affected by the shear. There is an optimal shear that you can have up to which the crystallization or the nucleation increases. Beyond that particular shear, it starts to decrease further, and if you increase the shear further, it might even degrade the system. So, for example, we have tried, instead of giving a syringe pump like this, we have tried putting a peristaltic pump for a continuous operation. But what we found is that in the peristaltic pump, as the pump basically grinds through the tubing, it basically makes a pressure wave and it pushes the liquid forward. But during that phase, there is a small region where it is squeezing the flexible tubing and pushing it forward, where the shear increases tremendously. The proteins start to denature at that particular point of time. So, even the operational wise, you are giving the same flow rate, but when you change the pump and the type of pump, the shear rate in which it is being pumped, all of these things might affect your crystallization, and this is one of the major aspects or challenges in that sense with respect to protein crystallization. In some ways, in some cases it might be good, because increasing the shear in some cases, as I told you, to a level, it might increase crystallization, but beyond that threshold it trouble. So, there is an optimization that you can, process optimization that you have to do in this sort of setup as well. Yes.
Sir, in cooling crystallization, initially we dissolve it in the solvent and then we cool it, right? So, after the protein crystallizes, how do we remove the solvent?
So, it depends on, say for example, in this case, in this particular case, it was a simple what is a filtration process at the end. So, you bring everything to the I mean, if it is a batch vessel, you just pour everything into a filtration unit and get the solids separated. Now, at this point of time, see, these are the image is not very clean here, but this is about 100 micrometers, and these are very fine crystals which are quite stable as well. And so, during filtration, as long as you do not give too much of mechanical forces into it, they do not break, or they are not say denaturing in that way. If you want, you could just add a little bit of buffer in order to wash those crystals as well, and then take it away, and means if you want to dry it further, you can put it in a vacuum chamber and dry it, and then you get very pure crystals out from it. So, that is one of the easiest way in a small scale. If you want to go into a larger scale, often what is often used is a centrifugal filtration units. You have the solution which comes directly into a centrifugal filter. So, because of the centrifugal action, the liquid means it would be covered with a perforated filter medium. So, the liquid get moved out from the filter unit, and the solids, that is the crystalline material, gets stayed back, and this can be scooped up or collected continuously or batch wise depending on your process requirements.
Is there any temperature range, sir, for this crystallization?
Yes, temperature, pH, your washing, all of these things can be regulated in order to, depending on what is your final requirement and how sensitive the protein is. See, for example, if you are concerned about any residual solvents which might be retained on that, you might have to change the solvent which is often used in the initial case, and then use a maybe slightly highly better volatile materials, or maybe remove the pH or change the buffers and so on, and wash the system. Again, temperature plays a big role, because if you are washing with too much of solvent, it might redissolve the proteins. Yes. So, again, while you are washing, you need to ensure that you are using a cold solvent in order to wash it. While drying, you do not want high temperature drying, right? For proteins, you cannot, they may not be, they might be thermally labile. So, which means that you have to do a vacuum drying at a lower temperature in order to make sure that the proteins are like not denatured and safe and so on. But one good thing is, once crystallized, they are much more stable. So, the stability range also increases for a crystalline material. So, something which might get easily denatured, let us say at 40 degrees, in a crystalline state, it might, the kinetics of the degradation might be lower. So, you can maybe quickly heat it up and then cool it down. So, these sort of manipulations in the process conditions can be done in order to clean it, in order to filter it and so on.
Sir, in case if we are doing freeze drying after crystallization, just we have removed the water using centrifugal separation, and then we are freeze drying the protein crystals what we have got. So, that freeze drying process will not again create some other crystals.
See, one thing about the crystallization process is, say looking at this aspect, this figure itself, let us say you cool it down up to this particular temperature, but your concentration never goes to 0. So, your mother liquor or the solution which is retaining after your crystallization or a cooling crystallization would still have some quantity of the protein that you want, as well as if you have buffer and other things, all of those things would be there in your solution. So, your solution is never going to be 100% of the protein is never going to crystallize out, which means that if you take this solution as such, including the leftover solution and the crystals and freeze dry it. Now, during the freeze drying process, mostly you would be removing only the solvent. All the salt, the buffer material, the uncrystallized protein, everything would be left out in the solution. During the freeze drying process, what would happen? All of this would form a solid chunk. It can be crystalline, it can be amorphous depending on how you do it, but then that would lead to a mixture of well formed crystals, which is that of protein, along with a mixture of salt, buffer and say crystallized protein, everything together, which would make the further separation much more difficult. So, if it is something which you can directly go with a freeze drying, and if it produces crystals during the freeze drying process, then you can skip the entire crystallization as well. You can directly go for this sort of freeze drying, but then you will never get this sort of large crystals, which, for example, as I told you, if you want this sort of inhalation patches for or patches for insulin, you want a particular specific crystal size, and it has to be crystalline, it should not be amorphous at all. So, these sort of properties it will not be able to match. So, that is one of the limitation of freeze drying, and of course, the scale in which you can do the freeze drying, it is always challenging, because you have to take the small vials in order to do even for large quantity, you have to divide into smaller vials and control it. But in a crystallization, if you can go with crystallization, you can scale up much more effectively. So, there are these ups and downs with respect to freeze drying versus crystallization and so on. Any other questions? So, this graph itself is a phase diagram in one way, because it is considering the solubility versus temperature, but there is when it comes to protein crystallization, temperature or the cooling crystallization alone is not the one which is often used. You use salt or precipitant along with proteins in order to precipitate or salt out the proteins in order to crystallize them. So, this is which is often used, which means that, let us say, if I am plotting the protein concentration against the salt concentration, I might get different zones. So, this zones are basically, say, this is the undersaturated solution where everything is in liquid phase. Above a particular concentration of the salt and protein, then you get a phase which can be in equilibrium between the solid and the liquid. So, this entire region over here, this metastable zone, nucleation and precipitation, all of this region represents a binary system, which is basically a solid which is in equilibrium with the liquid solution. So, the solid being the pure protein crystals that we are talking about. Now, when you are moving or when you are doing the process, let us say you are moving from an undersaturated solution system into an oversaturated solution or supersaturated solution, let us say you can do multiple ways. Let us say, as we have said earlier, if you are looking at the salt concentration, you can keep on adding a little bit more and more salt, so that you move from into a higher salt concentration region, or you can do evaporation and so on in order to reduce the solvent content and change the protein and as well as the salt content. So, all of these things can take you from one region of the phase diagram to the other regions, and typically speaking, you have got this metastable zone where the solid liquid equilibrium can exist, but formation of new solids or new crystals is generally does not happen, because what happens is in the steps of when you look at how crystallization occurs, it typically occurs in majorly you can define it as three different stages. One is the supersaturation generation, second is the crystal nucleation or birth of new crystals or the solid material, and the last is the crystal growth. So, the crystals which have grown or nucleated starts growing into a larger size crystal. So, first you have to generate the supersaturation, and then once a new crystal is born, then it starts growing, taking up the supersaturation from the solution, and it grows itself, reducing the concentration in the solution. So, this is what happens over here in this entire region. This is supersaturated, but as I told you, within this metastable zone, this is a region where it can be in equilibrium, but typically nucleation is absent in this, because there is not enough protein driving force or excess protein in order to drive the nucleation process. That is what happens is in the nucleation zone, if you are able to regulate your concentration within the nucleation zone, typically speaking, you might see spontaneous nucleation. So, if you are here within this zone, nucleation can occur, and as the nucleation occurs, it starts consuming the proteins. So, the protein concentration would fall down, which means that it might come down and move along this line vertically and move from the nucleation zone into the metastable zone, and once it reaches the solubility line, it stabilizes. once it reaches the solubility line, then it is in the solid form is in equilibrium with the liquid. So, then no further changes occur and it stabilizes at that point. Now, it is interesting to see that there is often in protein crystallization with salt, you see quite a lot of region which is nothing else but a precipitation zone. Now, what do you mean by precipitation? It is very low, low solubility, and the proteins or this molecules just start to precipitate and form the solid material. But now this solid material might not be crystals. These are not crystalline material. They are typically in the precipitation zone. What happens is kind of an uncontrolled conversion from the solid to the liquid phase to the solid form, and individual, it can be amorphous, it can be semi-crystalline, or it can be just like a denatured protein as well. So, it can even denature the protein and they might just precipitate out, which is practically useless as far as the crystallization perspective is concerned. In some cases, you might be able to take that precipitate, redissolve it, or give it the right conditions in order to remove the denaturation, or maybe allow it to get into its original conformations and then use it. But in many cases, it might not be always possible that once it is denatured, maybe it is very hard in order to bring it back into the actual system. So, this is something which we want to avoid in that sense. In most of the cases, we do not want to go into the precipitation zone. We want to somehow stay within the nucleation zone, nucleate a few crystals and then move into the metastable zone, so that the growth occurs and you get large crystals of your required size. But if you are able to regulate this in many ways, then you are practically trying to control the nucleation as well as the growth, then you can get the crystals of the required size, shape, or purity and so on. Now, the major challenge as far as the operational system operation of the crystallization is concerned is that we say that okay, it nucleates here, this is the nucleation zone, but nucleation is a highly stochastic or probabilistic phenomenon. In the sense, let us say you keep 10 nucleation or 10 systems to study at the same time, following the same conditions, maybe 2 of them crystallizes, you make sure that let us say all of them are within this nucleation zone. Even then, maybe 2 of them crystallizes, you see the crystals in 2 hours or half an hour, 2 others take maybe a few more hours in order to crystallize, 2-3 of them crystallizes overnight, by the next day you come, it crystallizes, a few of them might not even crystallize after a few days. So, when will it crystallize? Yes, thermodynamically it will crystallize, but how long will it take to crystallize? This is not something that you can easily predict or control. So, that becomes a very probabilistic event, which becomes again harder to control from the process operational perspective. In the sense, if you are using a batch vessel, how long do you wait for the crystals to appear? Means you are expecting in generally maybe in an hour the crystals to appear. In one batch it does not appear for 1 hour, you wait for another half an hour. How long will you have to wait? How long will you have to operate in order to get the same consistent quality? That becomes a big challenge since the nucleation becomes a highly probabilistic system. But of course, there are ways in order to control that. You can maybe in some cases you can even completely bypass the nucleation or the troubles of nucleation by adding seeds. The sense, let us say you already have pre-existing crystals maybe from the last batch of production. You take a very few tiny, very small quantity, small size of material from that and put it into this batch which is going to operate. As soon as it reaches let us say the metastable zone, you add the seeds into that, the pre-existing crystals into that, and now the good thing is you are in the metastable zone, so no new crystals would grow, only the crystals that you have added as the seeds, they will start growing. So that you can completely bypass the nucleation and get the process conditions right in order to get consistent quality of this, which is often used in the industrial scale. So, but on the lab scale or in the development scale, it becomes a big challenge where you cannot really control nucleation very effectively or thoroughly in that sense. Always it is a probabilistic process. Now, one of the most common aspect, especially when you are developing a new protein, is how do you figure out will it crystallize or not? Means if it crystallizes, under what condition it crystallizes, is it giving you the good crystals or is it just precipitating, etc. And in order to do that, as we discussed, there are so many different parameters. There is solvent, there is pH, there is salt concentration, there are so many of the temperature, so many different parameters to try. And how do you even like start? Where would you start? How do you design the experiment? That also becomes a big challenge. And more often, you are initially starting with very low quantity of protein to start with. You have got the chromatography, everything, you got the material, but how many, you do not have liters or even sometimes ml, you are working with microliters sometimes. So, you have very low quantity of the material to start with and large number of experiments to do. So, how will you go about it? How will you do if you are given a challenge for doing that? What is the first thing that you will do? Means, have you heard about design of experiments and so on? Yeah. So, you can use that sort of techniques in order to figure out, okay, what is the minimum number of experiments that you have to do. But at the same time, as I told you, means in a design of experiment, you are not going to trial each and every point in this phase diagram, and you do not know what the phase diagram looks like even. So, it becomes very difficult in order to even go ahead and design a conventional or use a conventional design of experiments approach in order to go ahead with that. So, but at the same time, see, this has been a long pending issue. So, what companies, what researchers have come with is development of screening protocols. There are set protocols that you can use, and many of them have gone into, let us say, automated systems with microliter scales. So, typically what you do is you have an array of let us say well plates, something like this. We will look into the well plate structure and so on in a little bit. I will just give you a brief glimpse of that. Now, you can run, means in order to identify a particular crystallization condition from a single system, you might have to run several tens or dozens of these sort of microplates, and each of them might be having a variation of, let us say, pH, salt or other conditions that you want to vary, and maybe within the same pH and salt variation you put one of them in at 5 degrees, the other one at 20 degrees, and see how it means how long it takes to crystallize or whether it crystallizes and so on. So, at different temperatures you need to do it. Some of them maybe you keep it mixing with some sort of mixing agitation action, or some of them you just keep it without any mixing so that it is static and see how it crystallizes. So, there are various things or various protocols which has been developed for this, and of course, when we think about developing it and doing it, means on paper it looks good. Let us say I give you 10 of these and ask you in order to set up all the 10 plates, how long it would take you to do this? Because you have to change the concentration of pH or the buffer, salt, everything in each well. It is not like, okay, you are going to pipette out the same composition into each of them. You have to vary, keep on varying between them. So, which is very difficult. So, that is why like nowadays researchers all over uses some sort of automated tools. There are multiple like injection profiles, vessels, everything that are useful. This is one such equipment which is often termed as like their name is mosquito, because of the nature in which it basically takes very small microliter of materials and put it into individual wells or vials. So, while the mosquito sucks out the blood from you, here it basically sucks in or basically adds a little bit of buffer, salt and proteins within the required concentrations and gradients at different wells. And these are all automated systems in order to make sure that, okay, like you would be able to screen as much as possible or protocols within a given period of time. And there has been so many developments in screening in the sense like we have been able to go into nano scale as well. So, you can see this is a complete nano well array, where in multiple procedures or in multiple stages you can add see proteins, buffer, precipitants, etc., with varying concentration, with varying conditions in each of them, and you maintain the systems at different conditions in order to get, let us say, full screening of the for entire phase diagram region. And we are going from, I mean, we started with milliliter scale experiments. Now the industry is going towards then when it on to microliter scale experiments. Now we are even at this range of nanoliter injections in order to do that, because as I told you, one of the major challenges is start with you do not have that much of quantity of protein to start. So, you have very little quantity of proteins to start with, then how do you even use that for a large number of hundreds or if not thousands of experiments to do with. And automated systems are becoming very common, like reducing the human interventions and so on, and making it much more accurate, including monitoring as well. The one which I showed you is only for just making the plates, but you have to take it out and monitor it. but nowadays there are entire platforms which are there, in the sense from one unit it starts filling in or making the plates, it moves to the next unit, it incubates for a given period of time, then it moves on to an imaging station where cameras take the images from multiple angles, maybe even using beyond the visible light as well, in order to see if there are crystals or precipitates and so on, and identify the crystalline or the conditions in which it crystallizes, and then move ahead, so that it basically automates the entire process, and the human intervention is limited to, let us say, initial feeding of the conditions that has to be checked, as well as what is the filling up of the buffers or the salt and the protein solutions and so on. So, there are quite a lot of industrially automated systems which are coming in, which can increase the throughput of the screening process. And of course, the next or the another major leap is on the predictive capabilities with the developments of the machine learning as well as the AI systems and so on. We are trying to bring in all these already existing knowledge, in the sense, okay, we know under what conditions already existing proteins do crystallize. Can you use that knowledge in order to develop, okay, means if you are given a new protein system, how would you know that it would crystallize, or how would you predict that under what conditions it is more likely to crystallize. So, these sort of data analysis, computational tools, machine learning based predictive models, etc., are coming in order to increase the screening capabilities, decrease the quantity of material that you are using for these processes, and make it more sustainable in the long run. So, these sort of changes are coming in. So, maybe we will take a quick 10-minute break here, and then recap after that, and move on with the protein crystallization methods after this. Okay. Thank you. Set of slides, we will see what are the methods which are often used for crystallization, and how we can do it both for okay, okay, fine. So, welcome back. So far we have seen what is protein crystallization, means what is the necessity for crystallizing proteins, and what are the phase diagrams, that is, and factors which are responsible, and also maybe into the a little bit into the screening. So, into the next slides, so we will start looking at the process of crystallizing, the what is the methods which is often used both for protein structure determination as well as for the purification and separation aspect. So, first and foremost, there are multiple ways for crystallizing proteins, and the primary classification is based on how do we generate the supersaturation in this one. Just a second. Excuse me. Yeah, fine. So, how do we generate the supersaturation? So, that is the primary objective that we would be first looking at. And as we have seen in the phase diagram, we have got the undersaturated region and the supersaturated region. So, the question is, how do we move from there undersaturated into the supersaturated as well as metastable and the nucleation salt? In order to do that, we can do it multiple ways. One, as I have discussed, how do we produce the salt? You evaporate the solvent out. So, as you evaporate the solvent, the concentration increases and gradually it moves into the supersaturated region. Once the initial crystals are formed, then that basically reduces the solution concentration and moves the system nearer to the solubility line. That is one way. The other one is cooling. We can reduce the temperature. Then we could also use anti-solvent or other materials which basically reduces the solubility without changing the temperature or evaporating the solvent. What we are going to do is we are going to add some other material which is fully miscible with the solution or the solvent that we are initially taking, and that basically helps in order to reduce the solubility. So, that is why it is called it reduces the solubility. So, that is why it is called as an anti-solvent system. The next one which is very common is this salting out approach, in which you add the salts into it, and these salts are basically there in order to modify your solubility or the product in which the solubility of the system in the solution, and without changing the pH or the other properties, it basically changes the activity of the solvent. So, maybe the water activity or the solution activity. Yeah, not sure why it basically changes. No, door. So, for the crystal structure determination, as we discussed earlier, primarily we work majorly on the microliter scale. So, the crystallizations can occur in microliter scale or in milliliter scales. Have you heard about this hanging drop method or sitting drop method and so on? Has anyone? No. So, these are often very commonly used screening techniques in protein crystallization. We could also have these sort of micro dialysis where membrane separation can also be utilized, or utilizing based on the interfacial diffusion and so on. In large scale processes, we will see a few examples of both batch, not batch we will skip, we will move into the continuous processing, how the crystallization can be done for a separation perspective. So, in hanging drop and sitting drop, as the name suggests, it basically depends on the orientation in which we are going to do the crystallization. It basically involves a small micro well inside which you would have, this is your buffer solution, which is called as the reservoir. It would have buffer and salts into it, and this is what is your protein solution which is going to crystallize. So, this is where you have got the protein. In addition to that, of course, you will be having other buffers and salts, everything as well in here. Now, what would happen if, let us say, if the concentration of salt is high in the reservoir and it is a bit low in the protein solution, what would happen over time? Any idea? Let us say if you just forget about the overall top solution, let us say you just take a little bit of, let us say, container with which is half filled with water and the rest is air, and you just close the system, what will happen? Will anything happen at all? That is, okay, and so the unless and until you change the temperature or the pressure, you do not expect any changes to occur, right? You do not expect any changes to occur. That is also because we assume that it would have already achieved the equilibrium. let us say to start with, maybe it is not in equilibrium, let us say the air there is maybe does not have any moisture in it. So, what would happen is it would need to achieve that equilibrium, some water might evaporate into the air, saturate the air. Once the air is saturated, then it would start condensing back as well, but nothing would be like macroscopically no other changes would be occurring, because there is the system would be in equilibrium, like 100 molecules are evaporating, 100 molecules are condensing. So, there is no major changes that you are. So, always the equilibriums are dynamic equilibriums, they are not static equilibriums. So, changes are occurring in the molecular level, but not in a macroscopic level. So, similar things would happen here as well. The liquid would start evaporating from the reservoir as well as from the protein solution, and it would also start condensing back after some time. Now, if you have got a higher solubility in higher concentration of salt or buffer or maybe other polymers in the reservoir, what would happen is both of the liquid means typically speaking the solvent used in both are the same, typically water. Water would start evaporating and it would basically start moving. There would be a net movement of water from the protein solution which is there upstairs on the top to the bottom reservoir. Simultaneously, there would also be a movement of water from the reservoir to the protein solution, but since we are going to have the system in such a way that the top solution have got additional protein and the lower solution would be having maybe having a higher amount of buffer or salt or some other components, the net equilibrium would be driven in such a way that the water would very slowly evaporate from the top droplet and would start moving to the reservoir. This would mean that over time the protein solution, the volume of the protein solution would keep decreasing in the hanging drop. Same thing happens in the sitting drop as well. There would be water which is evaporating from both the reservoir as well as from the drop, but due to the equilibrium gradual shift in the composition would take place, in the sense more water would evaporate from this droplet and less would condense back into it, whereas in the reservoir less would evaporate and more would come into it. So, overall the volume of the drop which is the sitting over there with the protein, it would keep on decreasing. So, as the volume of the protein solution decreases, what happens to the protein concentration? It would increase. Over time the protein concentration would increase, it would reach somehow to the nucleation zone and it would start nucleating and small crystals would be forming there. So, this is a very slow but effective in controlling the diffusion or the evaporation process and basically leading to a nucleation which can eventually produce the protein crystal. Now, why this is important? Because as the name suggests, this is a vapor diffusion method. Now, there is a slow evaporation which is taking place and slow condensation which is also taking place. Over time this volume change leads to a very slow change in the concentration. And the slow change is very much important because, as we have discussed earlier, if you in the zone of crystallization, the phase diagrams, we have precipitation zone as well as the nucleation zone and the metastable zone. So, you want the system to go into the metastable zone and into the nucleation zone, but you do not want the system to go beyond that into the precipitation zone. If it goes into the precipitation zone, everything would just precipitate out. But how much time do you need to give in the nucleation zone? Typically quite long, in the sense a few hours and or maybe sometimes a few days. So, it is which means that you have to evaporate it in such a low rate. That is where the diffusion becomes the controlling factor. So, by fine tuning the concentration of the materials in your reservoir as well as in the protein solution, by changing the salt concentration, by changing the buffer concentration, by changing the pH itself. So, all of these things can affect or change the vapor diffusion rate, and which can eventually lead to the formation of a what do you say, like very nice region, or that would lock the materials quite long time in the nucleation zone in order to ensure the proper nucleation of the protein. So, that is one of the conventional and the most reliable protocol which is often used for screening for various number of diffusions and the various number of conditions for a protein crystallization. And this, in order to make it scalable or in order to make sure that it can be automated and so on, we will see there are different types of wells. So, that is why I said we will come to the well structure later on. Depending on what is the type of experiment that you have to do, the well structures might also be different. It might be optimized for sitting drop, it might be optimized for hanging drop and so on. and typically speaking the volume of the components that you need to add here, this solution might be 1 microliter, 5 microliter and so on. It is not like really large. So, you only require few microliters in order to means if you have 100 microliters you can run a full plate with that. So, a full plate in the sense 96 different configurations can be run with that. So, there is a great tool in order to screen quite a lot of an area with a very small amount of material, and typically speaking this buffer, it might be 1 ml or 2 ml, that is again not much expensive, as well as it is not really difficult in order to make. So, that these buffer solutions can be added onto this region over here, again as I told you, with automated systems that can be added, and the hanging or the sitting drop can be used for that. Nowadays hanging drop is not much preferred. One of the challenges is that you need to first, how do you make the hanging drop? You first put the small droplet in a small cover slip, and then you invert it and put it on top of the valve. So, some sort of mechanical or human intervention is required in between, and some variations might result in that droplet spilling and so on. So, in order to avoid those sort of challenges, typically speaking sitting drop methods are sometimes preferred. Both have got their own drawbacks, because in the sitting drop method what happens is the protein crystal which is formed, it might get settled down at the bottom, and it might strongly attach a little bit to the bottom surface. Whereas on the hanging drop, the good thing is the crystal, because of its mass or gravity, it basically moves down and settles at the very bottom of your droplet. So, it will not be very strongly attached to the surface. So, the moment you want to take out, you can take out the cover slip and then easily remove the crystal, which would be very well formed and without any deformation. So, it gets more or less like a uniform surrounding system in order to grow. So, the growth would be much more uniform in a hanging drop method, but whereas in the sitting drop method, the bottom surface might be interacting with the crystal, that changes the shape and morphology and so on. So, that sometimes becomes a difficult task in order to retrieve the crystal from the sitting drop method. But means there are other ways as well in order to control the diffusion and ensure the small very small changes in concentration. One of the common method which is often used is called as microdialysis membranes, or you could even have a small oil layer on top of the droplet in order to reduce the evaporation rate, because if you have a little bit of droplet with a little bit of, let us say, surfactants or oil or something on top of it, on the top layer, that reduces the interfacial area that is available for the water molecules in order to evaporate. So, it might arrest most of the evaporation, but of course, even though we say that oil and water might not be miscible, still a little bit of very small quantity of water might be miscible into oil, and that quantity might mix with oil and evaporate from the other surface, leading to a very small rate of evaporation. So, that is also another way in order to control without going for these other techniques, or as mentioned, the microdialysis membrane, dialysis membranes which are selectively permeable only to the salts, let us say. So, only to the salts that was selectively permeable, that would also help in order to make the concentration gradients very gradual in order to give it enough time for the nucleation to happen. Other ways in order to control the crystallization is using gels. See, for example, you can have a tube kind of system over here, where let us say at the very top you add the protein solution, and the tube is filled with a gel, and at the very bottom you would have a concentrated salt solution. So, since it is a gel, means an agarose gel or something like that, which has got quite a lot of water content, but because of the gel structure and so on, the liquid does not really move as such. It is by a very slow diffusion that the liquid as well as the solid or the salt moves through that. So, that basically creates a very well defined concentration gradient over time within this tubing, and the proteins would start maybe moving in one direction, the salts moving in another direction. At some point, means within the same gel you would have various different concentrations. At one point you have got a particular concentration of protein and salt, at another point you are having another concentration of protein and salt. So, what happens is within the same gel or within the same column, you are now separating different positions within the phase diagram, because when you look at the phase diagram, at each point you have got different concentration of protein and salt. Now, that same concentrations, rather than changing it individually in individual wells, you are changing that continuously within a column. So, that can also be done, and by changing the concentration of the gel, how much of agarose is there, you can change the diffusion rates, You can use that in order to manipulate the growth rate or the nucleation rates, and say for example, in this case by changing the agarose concentration in the gel, you could produce very large crystals or very small crystals and so on. Now, keep in mind these crystallizations are not done for separation or purification as such, it is primarily done for growing good quality crystals for crystal structure data analysis. Because let us say you have got the crystals inside the gel, you if you are really looking at separating the protein, after that you will have to take the gel out, you have to dismantle the gel and get the protein out or the crystals out, which is going to be a tedious task if you are looking for a production perspective. But if you are only requiring 1 or 2 crystals for a crystal structure determination, then it makes sense, and you can also another aspect is you can identify various compositions within the gel and figure out at what concentration it crystallizes better, at what conditions it would nucleate faster, it would grow faster and so on. So, that sort of preliminary analysis can also be done, because typically speaking the diffusion rate of proteins as well as the salts can be figured out using experimental techniques, and that can be used in order to model the actual diffusion process and figure out at a particular time what is the concentration at a given position, and then has it crystallized there or not. So, then those sort of correlations can be done, which can be useful for developing the process conditions which are required for separation. So, here both the counter diffusion of salts and proteins occur, and as I told you can regulate the size of the systems. Now, once you let us say you have produced the crystals, how do you go about understanding the crystal size? So, crystal structure. So, what is the method which is used? I think we briefly discussed earlier. You have got the crystal, what do you do with that? Single crystal XRD. So, have anyone done or seen the single crystal XRD machine? We have. Okay. Data, have you seen in the sense? So, see it is basically a single component, single crystal that would be kept at the very center to which the X-rays would be shot at, and because see the X-rays are very small wavelength, they pass through the crystals, and what happens is the different atoms or molecules which are there, they diffract the crystals in different ways. Depending on the location or let us say the interferences, the diffraction pattern, etc., the X-rays which are passing through the crystals get diffracted and they are collected onto a sensor plate, which gives you an idea at what conditions or at what locations they are diffracting constructively as well as destructively, and this information can be correlated back into the position of these atoms. So, what typically happens is in the single crystal XRD, after your analysis you get a picture or image like this. This is the very basic data that you start. As you can see here, so at the very outset it might look some like random dots, but when you look at it, you might see some patterns emerging out from it. At specific intervals you see these sort of dots, and those dots correspond to regions where the X-rays are constructively interacting, and that leads to let us say specific gaps and intensities. Now, the intensity, the gap as well as the phase of the X-rays, the phase of the X-rays can be identified or needs to be correlated in order to go back and figure out what is the structure. So, there is a quite physical or involving quite a lot of computational or numerical analysis and so on. For simple molecules, salt crystals, etc., it is very simple, because you already know maybe sodium and chloride, these are the only 2 atoms which are there. So, some arrangements which would be very easy to figure out, but in case of proteins and large macromolecules, the trouble is you can't really just looking at it intuitively say, okay, this is going to be an FCC structure or this is going to be the unit cell dimensions and so on. You cannot make out based on that, but the fundamental theory and the Bragg's law, everything remains the same of how the molecules or the atoms interact with the see for example the X-rays and how it creates the diffraction pattern and so on. Based on that correlations as well as with additional knowledge on, for example, based on LCMS or based on other techniques, you might already have some idea about what is the primary structure of your protein, what is the amino acid sequences, or what is the composition of the different materials or amino acids which you are expecting in this. So, all of these informations can be correlated together in order to make the entire structure. So, after the initial data processing and phasing of the X-rays, which itself is a tougher another topic in itself, so we will not go into that, but what you generally get is what is the electron density what is the electron density map within that solid structure or the unit cell that you have that you are looking at. And based on that electron density map, you would try to build and fit what are the molecules within it. So, this could be something like a 3-dimensional electron density map that you can see, and as I told you, it is primarily giving you where the electrons are concentrated, and definitely which means that various atoms which are having different number of electrons would be having different signature densities and so on. So, those informations can be correlated, and in order to figure out what are the different molecules that might be there. So, these electron densities, once you figure out okay, what are the molecules, what are the structures which are occurring there, this would give you some sort of idea, okay, this might be the locations where this particular molecule is. Now, it does not give you directly the link between them or the bonds between them, but it indirectly gives you what is the spacing between this atom and this atom. Then you can work out whether they are directly linked to each other, and if you already have the one dimensional or basic amino acid sequence of that, then you know that okay, there is an amino acid of this particular nature. So, it must be having a particular carbamide bond, or it must be having a particular disulfide bond there. So, can you correlate with that and then expect means okay, what is the structure. So, all of this involves a number of iterations. So, it does not just go in a one single sequence, okay, you just get the diffraction, the electron density map, and then you just directly get the structure from there. It does not work that simple, but through a number of refinements and iterations you can get the idea about, pretty good idea about what is the structure in which the individual molecules or atoms are arranged. That can be then correlated in order to develop the entire electron map as well as the protein structure, and this gives you the final confirmed data regarding okay, this is for sure, this is the structure of that. And of course, the resolution and so on and other details matters depending on how well you are doing, how good the machine is, what temperature you are doing, etc. Because sometimes in order to get very good thorough knowledge, you need to cool it down to cryogenic temperatures in order to make sure that all the molecules are arrested in their position, and then do multiple XRD measurements in order to overcome the noises and so on. Even then, in most cases, let us say for example, you do not get that much of information regarding say for example, hydrogen. You in most often you do not see hydrogen in the simplest XRDs. Any can you think the reason why it could be? Exactly. So, it is the smallest one, and has only got one electron. So, here you are trying to figure out the electron density. So, the number of electrons do matter. So, more often this one single electron might not be good enough for you in order to get a confirmed information that okay, this is the exact position of hydrogen. So, that is generally an inferred information. So, more often you can figure out easily the carbon chains, other larger atoms and so on, it might be easier, but when it comes to the hydrogen and hydrogen bonding, etc., you do not really see that exactly in this protein structure determination through XRD, but once you have the rest of the chain which is sorted out, then based on molecular simulations, based on the orientations, etc., you can predict what could be the most likely configurations of hydrogen, and then based on that you can predict okay, is there a hydrogen bonding or not. So, what are the interactions, what is the van der Waals interactions, etc. Those kind of predictions can be done based on the inferences that you get from the XRD. So, that is how the XRD or the structural determination works, and as I told you, protein crystallization has been developed initially primarily for this particular purpose, in the sense how do you determine the crystal structure, and that is one of the limitation as well in the sense developing crystals, but this is one of the robust methods for determining the protein crystals, and it can be done for whenever you are developing a new protein where you do not have that much of information regarding the exact structure, its active sites and so on. This becomes a very good handy tool in order to understand, and so that you can then maybe you are trying to develop a new enzyme which can catalyze a particular reaction. You want a particular active site, you want to see if that active site is accessible from outside, you want to check what would be the configuration and so on. So, all of these kind of things can be understood better with an XRD system. Nowadays, there are predictive tools to some extent. Some of them are really said to be quite good as well, but means final confirmation, experimental confirmation can be obtained from the SCXRD system. Whether any doubts or anything?
Sir, it is said that the XRD data gives the spacing between the molecules, but with that spacing we cannot confirm the type of bonding, right?
See, again that is an inferred data, as I told you, because see, for example, if you are having a single bond between CC and of course including the nearby functional groups and so on, we can have calculations, molecular predictions, or even quantum calculations in order to figure out what could be the bond length between that, what is the interactions and so on, right. These can be correlated in order to say for sure or maybe infer whether it is going to be a double bond, whether it is going to be a triple bond, whether it is just a hydrogen bond. So, those sort of informations can be inferred, and let us say based on calculations we can come to a conclusions based on that. It is not just a guesswork in that sense, but quite a lot of calculations has to be done in order to thoroughly say that no, there is a bond and this is the structure and this is the location for
Sir,
that.
I am asking about this hydrogen bonding and which gives the folding, the secondary structure. The main reason why we are doing this single crystal XRD is we wanted to know the 3D structure of the protein. So, without knowing the hydrogen bonding or the ionic interactions or the disulfide linkages, how do we actually determine this 3D structure with this XRD?
See, that is why you get the means except for the hydrogen, as I told you,
Yeah.
you are able to pinpoint the location of the other atoms, other molecules. Once you have the individual atoms, so as I told you, it does not tell you okay there is this particular molecule there.
Yeah.
It gives you okay there is a chance for a carbon atom at this location. So, that is what looking at this one as well, you get a, say for example, you get a chain like this and you know that this is your amino acid sequence. So, you are expecting a chain or a like aromatic ring there or maybe a cyclic ring there. So, then you try to correlate with this and say that oh there is this aromatic ring, and then from there you keep on making the entire structure and the full peptide chain, let us say you are able to figure out. Once that backbone of the peptide chain has been figured out, then comes the challenge of your hydrogens. And again as I told you, you do not get exact pinpoint locations for hydrogens from the XRD in most cases. Nowadays there are refined techniques which have come which can give you better indications, but not again I would not say that they are not still as good as you are locating the carbon atoms and so on. So, what happens is after that it is based on your molecular predictions and simulations. Okay, you know that if there is a carbon here there should be a hydrogen. Now, in which orientation would the hydrogen be? Right, that is a question, that query that you would have. Now, based on previous experiences or databases that it has, what it does is if it is a non amino acid sequences and so on, then it would just place the hydrogens based on the previous ones, like previous non structures, and then see if the entire energy of the system is matching, if the interactions are matching, or is there any steric interactions, and all those other sort of calculations in order to rule out, let us say, what are the possibilities which are there and what are the possibilities that it cannot have. Now, once these are done, that is why I said it is an iterative process, just by the initial prediction or the guesswork it will not work. So, you need to do this iteratively to ensure that you get a minimized energy, minimized structure computationally, and then you try to relate this to the actual structure and figure out whether it is going to be stable. You can even correlate it to means macroscopic phenomena like the melting points or the denaturation, or let us say other bond energies, bond lengths and things like that. So, once you correlate all of these things, you get an inferred information regarding what is the exact position of hydrogen, what is the different distances between them, and these informations can be correlated to hydrogen bondings, van der Waals bondings. Let us say if you are having salt, or for example insulin, it crystallizes in the presence of zinc ions. The location of the zinc ion would be very easily determined through XRD because it is a much larger atom. Now, once you have the atom position which is identified and what is the nearby amino acids which are identified, then you can infer what is the going to be where the interaction is occurring, and that basically gives you a confidence. Okay, this is the location of my different atoms. Based on that, this is my zinc atom, this is my amino acid which is interacting with that. Now, I can compute and calculate what would be the bonding strength which is there, what can be the interactions, what can be the other hydrogen bondings and van der Waals interactions, everything there. So, these are all calculated and derived quantities from the XRD. Yeah. So, without the backbone you cannot even start where to figure out the hydrogen. Once you have the backbone you can go and calculate this. So, that is what the use is.
Basically, we will get the primary sequence first, then we will do the 3D structure using XRD, and then we will compute it to check whether everything is right.
Yes.
Is that the process?
That is the idealized process.
Yeah.
Now, there are cases in the sense like you do not have exact information regarding the primary chain, means sometimes it can also be done in such a way that, let us say, there are, especially nowadays with the modern developments, there are tools in which can, let us say, you do not have the exact information of this is the primary structure, there is a sequence of amino acids. You can still figure out the electron density map, try to correlate what are the atoms, what are the correspondingly what are the molecules involved, and then back correlate it to, okay, if I am having this sort of connections, this must be this particular amino acid. I can try to back calculate what is the primary sequence as well from the electron density map. So, that way it is possible, but again that would become iterative, and it would introduce more computational errors if you do not, if you are not doing the process very accurately, and that is why always having multiple informations together in order to correlate would be much better than only looking at one type of analytical tool. So, XRD has got its limitations, but at the same time once you have a concrete information regarding the backbone of the peptide chain, it gives you very good confirmation regarding what are the other interactions that you can calculate. So, that is why this becomes as the golden standard for figuring out what the structure is.
Thank you, sir.
So far what we have covered is basically how does the small scale crystallization works, and primarily focusing on the crystal structure determination. Now, when you think about in the large scale crystallization, the methods and the equipment etc., which are used might be quite different, because the target is now not to produce a single crystal which is of good quality or a particular size, but to produce as much as pure material is possible in the solid form. So, in batch crystallization we will quickly like just mention that once we have identified, let us say, under what condition it crystallizes, you can try to replicate the same pH buffer, let us say the same salt concentration etc., into a batch vessel by mixing the right quantity of materials. You can change the temperature if you want, you can cool it down and so on, you can use a heat transfer medium and so on in order to maintain the correct temperature or the cooling profile, and all of these things can be used for a regular batch crystallization. So, you can do it in 1 ml vessel or 50 ml vessel or 1 liter vessel. So, that can be done, and of course, as I mentioned earlier, shear can become a particular factor which you have to be worried about, because you do not want very high shear rate in typically in protein crystallization. So, you can use modified systems like the draft tube crystallizer. I believe you might have come across draft tube bioreactors, where you have got a central tube which basically channels you the overall flow in a more better axial along the axial mixing direction, and helps to reduce the shear and so on. So, those sort of configurations can be utilized. Now, of course, the challenge with batch processes are, they are of course batch. So, batch to batch variations are higher, your working volume is typically higher for a given product quantity that you have to produce. So, nowadays more and more of these continuous crystallization units are coming up, what you call as the conventional mixed solid mixed product removal crystallizers. That is nothing else but like a CSTR. You have got a bioreactor where you have got the central shaft for stirring. Same thing with slight modifications with temperature control and other modifications can be utilized for a single compound simple CSTR kind of crystallizer. You have nowadays with various flow crystallizers, it can be as I showed that image earlier, you can be a simple tube, let us say giving you a plug flow or a laminar flow inside, and that can be used as a for, you can cool it progressively or have different temperature zones, or have zones in which, let us say at one zone you add a little bit of salt, and then when it reaches the next zone you add maybe a little bit more of salt, or you change the pH by adding a little bit of an additional buffer. So, these sort of manipulations can be done in a single channel as the flow is occurring, and this can be used for crystallizing within the flow system. You could also have something which is called as the fluidized bed crystallizers, in the sense let us say you have the you have let us say a large reactor like this, and you provide the flow from the bottom, and the flow would be always moving up and then maybe coming down and recycled. But what happens is once the crystals are forming over here, because of the gravitational effect, because of the drag and so on, they would always remain fluidized. So, you can produce a solid bed of crystals which would, rather than settling down, which would always fluidize and get mixed within the system. and you can have maybe a different geometry at the top, so that let us say the velocity decreases, that changes the drag and so on, and that crystals settle down, and crystals would always remain within your zone of crystallization, whereas the solution can be taken out and recycled. So that you always have a what we call as a fluidized bed region where the crystallization takes place. So there are different configurations of continuous crystallizations that can be used in the large scale. Typically speaking, the batch processes, it can go anywhere between a few milliliters to several liters. And when you talk about continuous crystallizers, it is not the size of the reactor that matters, it is how much of material you can process in a unit time. So maybe you can process a few milliliters per minute, or maybe you can go with several liters per minute. So in a continuous process, there is called as throughput. So if you are able to process larger volume per unit time, then you are able to generate more material. So that way you can manipulate the system in order to produce as much as quantity that you want. So a few examples of continuous crystallization units, we will see, as I told you, it can go from a few microliters. See, this is a microliter scale microfluidic device. For example, here you get the protein and the precipitant, and you have got a carrier system which is coming, which is the oil, and this can produce smaller like droplets of proteins, and you can change the conditions or the temperature, etc., so that once it is mixed and produce a small kind of this droplet, it would crystallize after some time within this, and you can see that small crystals forming in most of these droplets. And when you look at the size, it is just 100 micrometer is the scale. So each of them is much less than 100 micrometer, which means the droplet itself is in the size range of your hair. So that is how small the droplets can be, and the crystals are growing inside there. So it might become a little bit tricky and troublesome engineering wise in order to recover them and purify them, but this is quite good for, again, good for protein crystal structure analysis and so on, and for large scale screening can also be done here. It can be done for larger processes as long as you are not requiring, see, liters to be processed in a short period of time, because it is only able to handle few microliters per minute or per hour in that sense. So there are many different varieties or variations of microfluidic devices which have been utilized for protein crystallization, and the good thing is you can have very precise control of the conditions, system, microscopic level control in order to get the crystallization done. On the other end of the spectrum, you have this sort of, for example, this is something which is called as an airlift crystallizer, which has been reported. So in the airlift crystallizer, what happens is, you have, similar to the fluidized bed, you are trying to maintain the crystals within this airlift zone. So you can provide air as the media which continuously moves up and provides the mixing, as well as it can help in order to maintain the required concentration gradient or thermal uniformity and the concentration uniformities and so on. So the mixing can be provided because of the air which is moving up, and the particles also might, or the crystals that are formed might get moved up and remain in a fluidized kind of system within the liquid system. And it can also be run continuously or batch wise depending on the requirement, and it can mimic, see, for example, this is a simple stirred overhead stirred crystallization system, and you can utilize that for protein crystallization. And you can see that typically speaking, the airlift crystallizers, because the shear is less, because the mixing is better and so on, in many ways they might be able to produce larger crystals, they might be able to produce well defined crystals that you want, sometimes better than the simple static mixers or overhead stirred mixers, etc., and so on. So the various configurations have come up. So I am just showing you a few examples from microliter scale to this is much larger scale, where you can have hundreds of mLs or maybe a liter of system which is operating continuously. Now the question is, how can you use it for a separation or continuous downstream processing for the production? Again, there has been many reported literatures based on that, since as I told you, my expertise is mainly on the pharmaceutical productions. This is one particular case, in case of antibody production which is produced. You can see that this is a 6 ml vessel, it can be scaled up to 100 ml and 1 liter vessels. The geometry looks pretty same, similar sort of mixing patterns and so on can be implemented, temperature profiles, etc., can be implemented, and you can see that during the crystallization process, the antibody concentration basically decreases from 10 gram per liter to practically around 1 gram per liter, which means that about 90% of the antibody which is there in the feed concentration, you are able to recover it as solids in the crystalline material. Of course, as I told you earlier as well, you cannot ever achieve 100% recovery, because there would be always some soluble component which is left out in the solution. So, that would be a loss, but you can try to minimize that quantity as much as possible, and you can see that this can be achieved within a very short period of time. Within 1 hour or 2 hours, you are able to complete the crystallization process. So, this is promising, looks promising as far as the downstream processing is concerned, but another challenge, as you might see over here, is you can see it is starting from 10 gram per liter, which might not be always a concentration which is achievable through a simple fermentation or a cell line production process. So, which means that some other primary separations might have to be done before that. And another aspect is when in the downstream processing, as it was discussed earlier, it is not always a single component of protein, there would be multiple proteins which would be present. So, when you have multiple proteins, how do you separate and segregate one protein crystallizes, the other one does not. So, this is another example where, for example, a mixture of lysozyme and tomatin has been selectively crystallized under different conditions. So, the blue regions, the green regions that you see here, they are conditions under which you can selectively crystallize only the lysozyme and not the tomatin, and this is the case where you can selectively crystallize tomatin without crystallizing the lysozyme, all from the same mixture. So, the starting mixture is the same, out from it you are selectively crystallizing one protein and removing the other. So, that let us say after the first crystallization you remove as much as lysozyme which is there, in the next stage of crystallization you can crystallize and remove as much as tomatin which is there. So, you can manipulate the process conditions in such a way that, or optimize the conditions in such a way that you can crystallize one after the other. Another approach which has been reported is using templates, in the sense proteins, as we discussed earlier, they are notoriously difficult to nucleate. So, you can support the nucleation, or you can enhance the nucleation by providing external heterogeneous templates. So, in this case what they have used is porous silica particles, or porous silica with specific size or nanometer range pores, and some of the proteins crystallize at one particular pore, and other proteins crystallize in the presence of other particular pores. So, these sort of pores or the heterogeneous templates can be used in order to enhance nucleation of one over the other. So, trypsin, for example, it nucleates if you are having the pores in the range of 3 to 4, whereas ferritin, it basically larger protein, it basically correlates at a larger one. So, these are again correlated back to the protein radius of gyration of the actual molecule of the large proteins. So, you can try to, if you have idea regarding how big the protein is, maybe you can design templates which can specifically figure out and crystallize only those particular proteins based on the size of the heterogeneous templates as well that you use. So, there are many selective methods which can be adopted. Another method is basically extraction plus crystallization. So, maybe one aspect is, once you have the cell line which is able to produce, let us say, monoclonal antibodies, you take that, you undertake maybe one or two processes of, let us say, liquid phase separations, maybe aqueous to phase extractions, or maybe other extraction methods, in which you selectively already, through methods you are basically after each precipitation or each extraction, you are selectively removing maybe half of the components, the next one you are reducing a few more, and after that it becomes a system in which maybe some of these precipitates can be again redissolved, or maybe taken back into another extraction medium, where the idea is progressively it becomes more and more pure. Once you have sufficient purity for the protein that you are looking for, you can take it up for a crystallization process, a conventional crystallization process like an evaporation, or maybe like a cooling crystallization or an anti-solvent crystallization, and that can be done. And again, going back to the phase diagram, you can figure out where you are starting with, how you can move, how do you change the concentration during the process, and basically how do you achieve the control over the crystallization. So, the process itself can be overlaid onto the phase diagrams in order to ensure that you get the right type and quantity of the crystals which is being produced. So, this is basically a case where, for example, you can directly use crystallization after a few stages of primary precipitations in order to generate about 98% yield of the target protein, and in this case it is a monoclonal antibody. Similar cases have been reported in order to overcome or eliminate or bypass the entire chromatography. So, rather than going for the conventional chromatographic based technique which involves all of these steps, you can directly take the output from the mammalian cell culture. You can use some pre-treatment, especially in order to precipitate out the unwanted proteins and so on, and you pass it through a membrane crystallizer, which is nothing else but selectively diffuses out some of the components and helps as a template for or concentrating the material, and that crystals which is formed during this particular continuous or batch process, it can be taken out and then further dissolved, and then finally you can directly take it for further formulation out from this, because rather than going for a multi-stage purification, you can go for a single stage purification, and as long as you are able to produce the crystals of the target protein, you can be assured that the protein or the solid material that you are going to produce is really pure. So, that is a major aspect of protein purification through crystallization, and of course, the primary challenges would be there in order to screen it and so on, but once you are able to achieve that, then it can become a cost effective process with minimal intermediate steps. Just to conclude with respect to the role in alternative protein, see, maybe it might not be applicable to each and every protein which you want to work with, and especially with the plant proteins or the mixture of proteins that you want to work with, it might not be really relevant in that sense. But on the other hand, let us say you have identified a few set of proteins, you want to study the structure, or maybe you want to take a particular protein and modify it further. Before that, you have to optimize and understand the structure and so on. So, in order to do all of these things, it might be helpful that you understand the structure through crystallography, and then you go ahead and manipulate the active sites and so on, and then let us say if it is feasible in order to crystallize, let us say you are producing enzyme for a particular application, or maybe you are producing a protein which has got a specific bioactivity, or maybe as a fortification agent, or say something of that sort, where it is a few different proteins which you have to really work with, then crystallization can be helpful in order to control or manipulate that specific or produce that particular proteins specifically, avoiding the rest of the mixture in your crude extract. So, it can be challenging, but at the same time this is an area which is worth looking at with respect to the future developments, especially when you are looking at products which want a long shelf life or which want much more purity, rather than looking only for the crude extract. If you can take and purify it, of course your market value also increases. So, in that sense it can be used, and just want to conclude overall. So, we have gone through the protein crystallization and how it can be, proteins can be crystallized, and what is the structure, how you can figure out the structure based on that, and we have also seen a little bit on the how do you do with the screening processes and the continuous as well as the batch procedures for crystallizing. And just to reiterate, it is more often valid and relevant when you want to produce specialized functional proteins and isolate it rather than producing the crudes. So, I would stop here, and if you have any questions or any queries, we can discuss that.
Sir, when we are extracting alternative proteins, the anti-nutritional factors also come with it.
The artificial?
Anti-nutritional factors and polyphenols will be extracted along with it. So, when we directly go for crystallization for purification, then won't the polyphenols or the anti-nutritional factors get crystallized along with it?
So, that is where this other method of prior extraction or precipitation might help, because let us say if you choose the initial precipitation or solvent extractions in such a way that it maybe removes the polyphenols or other materials, other major components at that stage, and increases the purity of the proteins a little bit, then it might be easier in order to crystallize the protein. Now, as I told you earlier as well, the good thing about proteins is if you are able to identify the conditions at which it can crystallize and you are able to control the process, then in most often the final product that you are going to produce is going to be pure protein crystals. So, the anti-nutritional matters might be there in the solution which you are going to start with. As long as their interactions can be changed or manipulated in such a way that it is only the proteins which crystallize, the solid material that you are going to get more likely is going to be pure proteins, excluding most of these anti-nutritional matters and other proteins as well. So, this is why it can act as a single step purification, because rather than going with the number of chromatography columns and sequential purification, this might help you in order to get that crystals out. Once it is a crystalline material, then you can be assured that it is pure and stable in that sense.
So for that we have to get the particular crystallization condition for a particular protein.
Exactly. So that screening becomes much more important there. So
Okay.
based on the specific material that you are starting with, based on the protein that you are targeting, that screening for the process conditions, that becomes challenging, or that becomes the more time consuming, but once that is established, then you can be assured that you can always produce the right crystals.
I have one more question, sir.
Yes.
The protein polyphenol interactions are very strong. So will crystallization be able to overcome that?
Say, means as mentioned earlier, in some cases the proteins, they do crystallize not individually as proteins along with the stabilizers or other interacting components.
Yes.
So, if the polyphenol, means if you are not able to break the interactions, let us say by changing the pH or salt concentrations, etc., that is where, as I mentioned, PEG and things like that might help, because adding these sort of polymers, what happens is they change the activity coefficients of the solvent, they change the activity or the interactions between these other stabilizing components and so on. If you are able to disrupt that polyphenols protein interaction, then that is really good. But in some cases it so happens that the proteins crystallize better with these interacting factors. So, see, there is no hard and fast or a universal answer for that. In some cases, ideally speaking, you want to remove the everything else, polyphenols and so on, and to crystallize this alone. You might be able to, if you work enough screening and so on, you might be able to identify the correct pH, like salt concentration, all of those conditions in which you can disrupt that. But it is not always assured, and let us say if it is crystallizing with the polyphenols, let us say, then it sometimes it is easier to do it that way. You take the you do the crystallization, you get the solids. At least one thing that you can be sure is, now the mixture that you are going to get is only containing that your protein and only maybe one or two type of polyphenols. You cannot have so many different types of materials coming into the crystalline structure. So the purity of your separation, like crystals, is going to be much higher than the material that you started with.
Okay.
At least you can reduce quite a lot of impure materials through that. So that is one thing that you can look into.
Thank you, sir.
Any other questions? Okay, that's all. Thank you. Thank you for your questions and interactions as well. And at any point of time, if you feel you need to clarify anything and so on, you can contact me. So this is my email ID. So you can contact us if you need any help with the crystallization process at any point of time. So as I told you earlier, I do not work extensively with protein structure determination as of now, but we can help with the crystallization aspects if it is required. Okay then, thank you all.